the next section of the program, which is the, let's see, it's the lightning sessions. And the way that this is going to operate here is that these were the instructions. Everybody is limited to 3 slides. We'll see how many of you stuck to the 3 slides. And then you will have 2 minutes. I will introduce each person and I will read your title. So if I read your title, no need for you to do it. So our first speaker is Ranvar Tung. He's a student from Fresno State, and then the title of his slide is talk is plant physiological responses to different salinity levels and young pistachios planted in filled lysimeters. So Ranvar, go ahead.
All right, could I get the next slide? All right, so a brief intro. A brief intro about this is pistachios are known to be somewhat tolerant of salinity. And so there are many pistachio plantings in saline conditions. An example of that is the west side of Fresno County. So along the I-5, you're going to find a lot of large acreages of pistachios. And in addition to this, bearing pistachio acreage has more than doubled in the past decade. It has reached 264,000 bearing acres in 2018. So with this increasing acreage in saline soils, it has called for more research into the salinity tolerance of pistachios. So our experiment is a randomized complete block design for salinity treatments to rootstocks. The salinity treatments are 0.5 decisiemens, 4 decisiemens, 8 decisiemens, and 12 decisiemens. And the EC is comprised of sodium chloride, 6 parts per million boron, and 1 part per million selenium. And the 2 rootstocks are UCB1 and PG1. So just some overview of the data that we've analyzed so far for the season. From the Julian date 150, we've seen the leaf area significantly decrease, and we believe this is due to the salinity treatments. Something that we've noticed is the chlorophyll content has increased, and we witnessed a drastic increase on Julian date 282. So it's something that we need to look into more and sort of figure out why it happened. And in general, when it comes to the total growth of the rootstocks, UCB1 has shown a higher growth rate than PG1 in roughly all of the salinity categories. And that's it.
My gosh, Ranvar, you did very good. That was less than 2 minutes, so excellent. So our next speaker is Darren San. He's a student from San Luis Obispo. The title of his is Nitrification of Flush, Dairy, Lagoon, Water, and Biofilm Reactors. Darren. Oh, Darren, there we go.
Yeah, hello, thank you. All right, so to start us off, dairy waste is an inevitable.
I think you, let me just interrupt. Can you speak up a little bit more? There we go.
Yeah. Hello.
There we go.
Can you hear me?
Yeah, but barely.
Oh no. Okay, is this better?
It is better, thank you.
Okay, sorry. All right, so dairy waste is an inevitable issue for all dairies, and existing treatment solutions are expensive and high maintenance. At Cal Poly, our dairy unit manages their dairy waste, specifically cow manure wastewater, with a freshwater system and a storage lagoon. And so ideally, these lagoons would be able to treat ammonium using bacterial processes, but realistically, lagoons lack the oxygen needed to convert ammonium into nitrate in that first part of the bacterial process. And so existing treatment solutions include aeration. However, again, these are high cost, high maintenance, and nitrification is not always guaranteed with these with just aeration. Next slide, please. All right, so for my thesis, I investigated algae raceway ponds. These ponds are equipped with paddle wheels for mixing, and they grow algae, which photosynthesize and naturally produce oxygen needed for nitrification. And so as brown as our ponds were, we did have algae, and we also saw nitrification byproducts. And so we saw nitrification happening. Comparing the summer and winter performance, we ultimately came to the conclusion that ponds should be designed for winter conditions where sunlight and temperature can have a greater effect on photosynthesis and thus nitrification. Next slide, please. Yes, so for my second pilot treatment system, we investigated aerated biofilm reactors. These are 2 tanks in series, each tank equipped with an aerator and a cartridge of geotextiles for biofilm to grow on. And so we tested 2 types of geotextiles. We tested a smooth bonded geotextile and a fuzzy non-bonded geotextile. And we suspected that the fuzzy geotextile would have greater surface area for bacteria to grow on. And we did see that. We saw that there was more attached biomass on the fuzzy geotextile during batch mode. However, when they were continuously operating, they also accumulated more sludge a lot faster, and that impacted the amount of ammonia that was removed from the wastewater. We also conducted a scrape test and found that the biomass suspended in the water was 10 times greater than what was attached to the biomass. And so ultimately, with our future research, we'll be investigating the effects of regularly removing sludge and the effects of attached biomass compared to what's suspended in the water. Thank you.
Okay, thank you, Darren. Our next speaker is Nancy Buckley. She's faculty Cal Poly Pomona, and she'll be speaking about investigating the effects of garlic on macrophage response to microbial agents, but it looks like maybe we have a student. Nancy, or if, I'm sorry, go ahead.
Yes, can you hear me? I can't turn on my video, but I can turn on my voice. So I'll be presenting, but this work was largely done by 2 of my undergrad students, Eric Lee and Benjamin Soto, who are now in grad school. And we are investigating the effects of garlic on the macrophage response to microbial agents. And in the next slide, we can see that macrophages that we used, which are one of the main cells in the innate immune system, were either from cell lines that we purchased or macrophages. Next, please. We can unanimate this slide. Or macrophages derived from mice. And these macrophages were then treated with either the microbial agents, Candida albicans. Can you keep on clicking, please? Thank you. And 1 more click, 2 more clicks. Microbial agents, Candida albicans, or LPS derived from E. coli, and garlic. And what we were measuring was a very important pro-inflammatory cytokine, tumor necrosis factor alpha, which is majorly produced by macrophages. Next slide, please. And what we found was very interesting in that garlic is able to modulate this very important pro-inflammatory cytokine in these macrophages. And as you can see here, we used either mice derived from female F or male mice, or the cell lines J7 and raw cells. And we saw that in each case, the white bars represent the microbial agent by itself, and the black bar represents the microbial agent plus garlic. When the cells were treated with garlic and the microbial agent, there was a suppression in that pro-inflammatory cytokine. And this is consistent with what has been coined for garlic, which is suppression of inflammation. However, the interesting part is that on the right-hand panel, we see that when the cells were treated with lipopolysaccharide derived from E. coli, the tumor necrosis factor alpha pro-inflammatory cytokine was not suppressed, but instead was stimulated. And we were using exactly the same cell lines that we used for the previous study. So we, and the most important thing that we found is that this effect of garlic was cell-specific, as 3 of the 4 cell lines used were not behaving the same. And you can see here that the raw cells, the last 2 bars, are not having, the garlic is not having an effect on that cell line. So then subsequently, we looked at cell concentration. 1 more click, please. And we see that when we compare different cell concentrations between the J7s and raw cells, we saw that although garlic still stimulated TNF-alpha production at all cell concentrations in the J7 cells and the raw cells, this was very dependent on the concentration of the cells. So in conclusion then, we found that garlic is able to modulate this very important pro-inflammatory cytokine in a cell-dependent, concentration-dependent manner, and also depending on the microbial agent with which the cells were challenged. thank you very much for listening. That's all we have.
Okay, thanks a lot, Nancy. Our next speaker is Subodh Bhandari. He's a faculty member at Pomona, Cal Poly Pomona. The title of his talk is Towards Determining Nitrogen and Water Threshold for Lettuce Production Using Agronomic Measurements and Remote Sensing Data. Subodh.
Good morning. So our primary goal of the project is using UAV-based remote sensing technology for precision agriculture, and we are also investigating in determining the effectiveness of remote sensing data for as well as the agronomic measurements for determining nitrogen and water threshold for lettuce production as well as other plants. So for the experiment design, what we are doing is we have a lettuce plot and we are varying the amount of nitrogen and irrigation from 0 to 100% at an interval of 25%. And the data we are collecting are agronomic measurements such as leaf numbers, plant height, and the UAV remote sensing data. So we have two different UAV platforms, fixed wing and multicopter. One is equipped with a multispectral sensor, and the second one with the hybrid spectral sensor. So next slide, please. So what we have found is from the data collected over several periods, growing seasons, the effect of irrigation on the left-hand side, the number of leaf numbers. So that is with the 50% and 100% nitrogen, we have a similar number of leaves. Similarly, on the middle, that is the effect of the nitrogen treatment. Also, for the 50% and 100% nitrogen, we have similar numbers of leaves. And we also looked at the total lettuce production yield, and as we see here, the 50% nitrogen and 100% nitrogen, they have similar values. And then next, what we looked at is the effect of the remote sensing data. So the graph on the bottom left is the water band index. We compared that with the water content on the lettuce leaves, and we found out that the correlation so far is 0.48. And another index, widely used index that we used was the normalized difference vegetation index. We compared that with the nitrogen content. However, the nitrogen, the correlation with the nitrogen content has not been found very good so far. However, the chlorophyll content has a very good correlation, or the better, significantly better correlation with NDVI. So that is, we have gotten up to 0.7. With the nitrogen, we are continuing looking at nitrogen, why we have this result. And one of the things is we have limited data, as we have to send these samples to Nebraska, that is for in a lab for the nitrogen content determination. But we will continue doing that. So right now what we are doing is working on investigating the accuracy of, or increasing the accuracy of remote sensing data. So with that, hopefully we'll get better results. That's all.
Thank you, Subodh. In the future, what I'm going to do is at 2 minutes, I'm just going to jump in with my mic and say 2 minutes just to give everybody a heads up to wind it up. So our next speaker is Cindy Daly, a faculty member at Chico State. The title of her talk is Impact of Conservation, Tillage Strategies on Organic Vegetable Yield and Implications for Nutrient Density. Cindy.
Great. Thank you so much, David. Appreciate the time. And we're going to go on to the next slide because I promise this will be 2 minutes. So next. Most of the literature shows that no-till improves soil organic matter. And as you improve soil organic matter, you improve water holding capacity. And we see that no-till systems also sequester lots of CO2, improve soil aggregate stability, reducing erosion, and so on. Next slide. And even with everything that we know about the no-till systems, tillage still continues to be one of the major forms of weed control in many cropping systems, but particularly in vegetable cropping systems next. So the objective of our project is to look at the cost benefit of a no-till organic veg production system by comparing soil quality, yield, and cost of production. And we also want to tie this back to nutrient density because we think that there's a story to be told there as well with the impact that it has on nutrient density of crops. Next slide. Our preliminary data, this is basically our cooperators for the study. We do have 5 locations throughout the Central Valley, including the OVP at the University Farm in Chico. Next slide. Our preliminary data is kind of interesting and it shows the yield drag. And that's not uncommon. We see that in the literature and our cooperators have seen the same thing. So we are seeing a significant drop in survival of our transplants in peppers, and we also saw a drop in yield, significantly so in eggplant. So, that's not, that doesn't speak very well for the no-till system. As we move into our next crop rotation, so next slide, we no longer have the yield drag. So we see that there is actually an improvement in overall survival rates in our garlic. Next slide. We're still working on our soil health data, and we think that a lot of the story can be told here with respect to what's going on in the soil with percent carbon, percent nitrogen, aggregate stability, and so on. But that data is yet to be seen. We are seeing a slight increase in our active carbon levels. Next slide. And if you look at our overall nutrient density, this is just our peppers. So again, preliminary data, but we are seeing that there is an improvement in polyphenol concentration in our no-till system, presumably due to the change in the biological microbiome in the soil itself. Next slide. So that's the conclusion of my 2-minute, and I hope I'm under, David, am I?
Well, just very close. So thank you, Cindy. Good job. So all of the panelists, all of the people that are speaking today, we will have a few minutes left over at the end for asking questions. And Cindy, I've got one for you, but I'll hold it. So our next speaker is Samarjeet Singh. He's a student at Fresno State University. And the title is, and then also if you can turn on your camera, please, Samarjeet. Modeling approaches to spatial analyses of soil salinity in a small plot experiment. How did it become so complicated?
Well,
Go ahead.
Everybody, I can actually start the video actually, but I can turn off the microphone. So hey everyone, so today's title of my presentation is the modeling approaches for spatial analysis of the soil salinity in a small plot experiment. As we all know that soil salinity is one of the major abiotic stresses all around the globe, contributing to salt accumulation in the farmland due to certain factors such as less precipitation, evapotranspiration, and shallow groundwater. And due to such region, forest land in our Central Valley is being cut down in order to reserve farm resources for more profitable crops. California is #1 wheat producer, and alfalfa is the first choice of our Western ranchers. So that is a reason we need to thrive our forest industry. So we're in this experiment, we're evaluating 35 new salt-bred alfalfa cultivars in clay soil under subsurface drip irrigation in the Western San Joaquin Valley. So these problems of spatial and temporal variability in the soil profile certain times leads to ineffective variety comparison and raises the further problem in the variety selection for farmers. That is one of the basic and important selection for the farmers. So our objective here is to assess the uniformity of the soil salinity imposed by 2 of our treatments. One of them is low saline irrigation, and the other one is high saline irrigation. Next slide, please. So just to give you an idea about our treatment, in the top chart, you can see our low saline treatment is around 2 decisiemens per meter, and whereas our high saline conditions are anywhere from like 8 and going up to 13 decisiemens per meter. And as you can see in the below left picture, these are the 4 basins, number 1 and number 3. As we can see, certain bare soil patches as due to the salt stress, and number 2 and 4 are the low saline conditions. So there are various geophysical methods such as electromagnetic induction that can be effective for monitoring changes in soil salinity. And one of them is the EM38 MK2 sensor. That is what we're using in our case.
Okay, that's 2 minutes. So let's keep
So,
going.
so basically we have done like 5 surveys, 1 in the end of last year and 4 this year. So right now, next slide, please. Right now we're in the process of modeling the ECA that we get from the EM38 and convert into ECA, that is the actual amount of salts in the soil. So right now we are seeing certain trends that are observed that may cause variation within the variety trial. So our final, the spatial and temporal trend of soil salinity will help decrease the error within the variety difference, serving as a covariate. That's what we're doing right now. And thank you all. That's it.
Very good. Thank you.
Sorry for that.
No, not a problem. Thank you so much. Great correlation there on your model there.
Yeah, 96.
Yeah, our next speaker is Armando Arturo Vega Orsorno. He is a student at San Luis Obispo. The title of his talk is Chemical and Sensory Effects of Co-Fermentation, Post-Alcoholic and Post-Malolactic Blending of Malbec, Merlot, and Petit Syrah wines. Armando.
Thanks, David. I'm trying to set up my video, but I can't start it because the host has stopped it.
Oh, is that right? I will see if I can fix that somehow.
Thanks, David.
But I'm thinking you're going to have to go ahead because I'm not going to make any promises that I
got it right now.
Yeah.
Okay.
Now I can see you, so I think you may be on.
Yeah, I'm on. Thanks. So, you know, the California wine industry is extremely relevant, economically speaking. It produces 90% of the wine in the US and it employs thousands of people. At the same time, there is a consumer drive towards wines with fewer additives and less intervention. So what is co-fermentation? Co-fermentation is a winemaking technique that entails mixing of 2 or more grape varieties at the moment of crushing. Co-fermentation delivers wines that exhibit or that seem to exhibit more complexity compared to wines that are blended post-alcoholic or post-malolactic fermentation. So red wines contain 2 families of compounds that are especially important. These are anthocyanins and tannins. Anthocyanins are responsible for color, and tannins are responsible for the grapey mouthfeel. So my research consists of comparing how varieties or how wines that are co-fermented will contrast to wines that are blended post-fermentation. I'm working with Merlot, which has medium color and high tannins, Malbec that has high color and low tannins, and Petit Syrah that has high everything. So next slide. I think this is the previous presentation because there's more than 3 slides. But anyways, if you could just go to the one with the graphs, that would be nice. Thank you. Yeah, this one has like 20. So.
Oh, gee.
It's gonna be a while. That's my last one. Okay. So after following the winemaking process, we utilize humans as sensory instruments in the sensory panel. We make sure the panelists are blinded to the study, so they don't know which wines they're being fed to. And at the end, we pull all the results and we assess with statistical magic to see if the end consumer can really make a difference between the co-fermented wines versus the post-blended wines. In these plots, we can see the co-fermented wines exhibit different characteristics from the blended wines. And they also exhibit a long-lasting color too, which is due to polymeric pigments. These compounds are formed between anthocyanins and tannins. So if you could go to the last slide, whatever that might be, that presentation. So co-fermentation is important because we can construct more complex wines with less intervention, which is a pretty relevant trend nowadays. So if you could just head to the last slide. I would like to thank my advisor, Dr. Xasa, the CSU REI, and all the migrant laborers that made California wine possible. Thanks.
Thank you, Armando. And I like the part where you said we're just going to use some statistical magic.
That's what it is, yeah.
Yeah, indeed. So our
Yeah.
next speaker is Dilruba Yisman. She's faculty at Fresno State University. The title of her talk is Almond Water Stress and Yield Analysis Through Soil Plant and Image-Based Technologies for Sustainable Almond Production. Dilruba. Unmute your mic. There we go.
All right. Thank you, David. So almond is one of the most vibrant cash crop in California's economy, but it uses about 8% of total allocated water for agriculture statewide. So, cost-wise, almond is costly to produce if we think about water. Now, keeping that in mind, this project was targeted to, examine if water use in almond can be minimized without sacrificing yield by utilizing different kind of, precision ag technologies such as sensors, imageries, and also other fieldable measurements. Next slide, please. So for this particular study, we introduced two different intentional water reduction treatments, 85% ET, irrigating at 85% ET, which is 15% water reduction, then irrigating at 70% ET, which is 30% water reduction, and then irrigating at 100% ET. That means no cutting, no water, 0% water reduction. So these three treatments are replicated 4 times in the orchard located at University Agricultural Laboratory at Fresno State, and all of these treatment plots were located in the same soil type, which is Delhi Lumi sand, pretty excessively drained sandy soil. We installed different, sensors like, you can see in the right picture, soil moisture sensors with a remote data logging system. Then next slide, please. And then we try to take real-time data at 2 different soil depths, 6 inches and 18 inches. We took stem water potential data biweekly, and also we collected imagery data in the form of NDVI only, and not the common index, vegetation index, and then finally we collected yield data too. This is a continuing project, so we still are taking data for this project. This particular orchard is 12-year-old, we butane Padri 2 almond varieties. So after 2 years, the first yield data shows some results that I would like to present here. As you can see, for these 2 particular varieties, on an average, 65 pounds per tree almond production is normal. So in Butte variety, we saw that it shows less sensitivity to water reduction than Padre variety. So for Butte variety, even not cutting any water gives us 54 pounds, but cutting 15% of water gives us even more yield, 57 pounds. And for 30% water reduction, it goes down a little bit, but not too bad. For Padre variety, the trend is completely different. It is really sensitive to water reduction, 10 pounds less every 15% water reduction. So this is kind of like the preliminary result, but we are kind of looking closely with all those monitoring data, and also we'll be taking more yield data moving forward so that we can make a good conclusion if it is actually variety specific, minimizing water or not. And that's pretty much it. Thank you.
Okay, Dilruba, thank you. That's some great looking data, by the way. Our next speaker is Sashika. Let's see, let me see, Sashika, let me see if you can turn your camera on. Sashika.
Hi, David.
Yeah, I'll fix you here in just a minute.
Okay.
And the title of her talk is Enhanced Integration of Verticillium Wilt Management Practices in Strawberry. Go ahead and get started, and then we'll try to get your camera up.
Good morning, everyone. Thank you, David, for the introduction. Click, please. In this photo, you can see the disease symptoms of yellowing and stunting and plant mortality. Next, click, please. In here, you can see the disease cycle. The pathogen reside in dead plant material and soil as these black hardened structures called microsclerotia. When the strawberry transplants establish in soil, the new roots exert compounds that can act as a cue for the microsclerotia to germinate. Click twice, please. Our goal is to break the disease cycle at 2 points to prevent the pathogen from making microsclerotia and also to reduce the soil-borne microsclerotia. Our team includes myself, Cal Poly Strawberry Center Director, Dr. Gerald Holmes, Assistant Professor, Dr. Chanping Ding, graduate student Jack Koster, and undergraduate student Elias Periga Hernandez. Next slide, please. This is our experimental design. We have 3 treatments: a crop termination, bed fumigation, and combination of treatments with 3 fumigants and 2 cultivars. Next slide, please. We will grow a susceptible cultivar of strawberry at our Cal Poly strawberry field this fall and monitor the disease progression, conduct the treatments next summer, plant susceptible and resistant cultivars next fall, and monitor the disease progression, get the yield assessments till summer of 2022. We have so far done the first 2 steps. And with that, I would like to thank you all and take any questions at the end.
Yeah, thank you, Sashika. We will save all of the questions for the end, and then we can ask all of the panelists. So thank you for doing that. Our next speaker is Alex John. He's faculty at Pomona, Cal Poly Pomona. The title of his talk is Making Chemicals from Agricultural Waste. Alex?
Thank you, David, and good morning, everyone. Let's go ahead and do 3 clicks for this slide here. So I guess my talk is sort of different from whatever we've been hearing because my research is focused on addressing a sustainability problem, and people might wonder, you know, how is this related to agriculture? What we want to do is we want to find an alternate source for making our products actually. So the materials that we use. So these could be, as I've shown it on this slide, it could be adhesives, you know, it could be plastics, it could be any other material, even like the clothes that we use. And so these are all currently sourced from petroleum, from crude oil in some sense. And so what we would like to do is we would like to go for an alternate source. And since these are all materials, we need chemicals, that's carbon, to make these compounds. And so a renewable source of carbon is basically agricultural residue. So can I get another click here, please? Yes. So agriculture now, since California is a big agricultural state, we have a lot of agricultural residue here. So we don't want like, I mean, we're just talking about like all the waste that's generated. It could be like tree prunings, it could be like shells, it could be anything else, forest residues, all of that. And California does have a lot of it. And the other good thing is this is an industry that generates a lot of jobs. So the most recent report that came out that suggests that it generated about 4.65 million jobs and added about 500 billions to the economy. Let's go ahead and go for the next slide. So what we're doing here is we're trying to like basically convert lignin, which is a bio-based polymer, into smaller chemicals. So our approach here is to generate a library of lignin-like molecules, smaller molecules that resemble lignin, and then study vanadium catalysts for converting them into smaller molecules and then eventually develop structure-activity relationships. Can we go to the next slide, please? And 4 clicks on this slide, please. So these are some of our preliminary results where we started working on some lignin model compounds. Especially we're using this beta-O4 model compound here, which can be cleaved into compounds like benzoic acid and phenol, benzaldehyde, other things. And so we use NMR to follow a lot of our reactions, and we've synthesized a lot of these catalysts that you can see that are numbered here like 3A to 3F. And just to draw your attention to like maybe 2 of them, if you look at entry number 1 for 3A and entry number 4 for 3D, what you notice is changing that catalyst structure actually changes the yield. And so that's the sort of data that we would like to generate as part of this product, as part of this project. So thank you. We can go on to the next slide, which is the acknowledgement slide. So I want to thank my research group, which does a lot of this work. Thank you.
Alex, thank you. Very succinct, very good job. Very interesting stuff. We'll have questions for you here at the end for sure.
Thank you.
Our next speaker is Lisa Bentley. She's faculty at Sonoma State. The title of her talk is Quantifying Above Ground Carbon Stocks and Biofuels Across Diverse Gradients to Inform Natural Resource Management in Light of Climate Change. Lisa.
Thanks, David. So this project is a collaboration with the people and organizations who you just saw in that first slide. And I'm here to explain why we want to do this in light of agriculture. So as some of you might know, the California Air Resource Board's Natural and Lands Working Inventory provides a quantitative estimate of carbon stored in the state's land base. 85% of the biomass is found in forest and shrublands, but there's 20 to 40% uncertainty in these estimates. So since the National Working Lands Inventory plays a very important role in the state's climate strategy, by contributing to carbon sequestration and greenhouse gas reduction, it's a key tool to estimate the carbon stored in timber, and we have to reduce the uncertainty in these estimates. So in addition, as you can see on this picture, the increased intensity and occurrence of wildfire in California leads to a buildup of fuels. And so as a result, the landowners and managers need to evaluate fuels management approaches to increase forest health, which requires this accurate quantification of fuels. So you can go to the next slide. So hopefully I've convinced you that there's a need to rapidly and more accurately estimate above ground biomass and fuels parameters. As such, the objective of our work is to use a terrestrial laser scanner and a drone combined with modern data processing techniques to acquire detailed measurements of three-dimensional forest structure. We're collecting these data across 5 sites in Northern California. We've sampled about 99 plots to date. Our aim is to collect data both before and after fuels management or wildfire to evaluate both the effect of treatments on above ground biomass in timber and fire fuels. Next slide. And so right now we're in the data analysis stage with both the drone and the TLS data. So the TLS data are shown in this video, which might be a little bit laggy, but hopefully you get a chance to see some of it. And we're really hopeful about these data, especially the utility of the TLS data, because once we extract these individual trees, which is shown in this point cloud on the upper right, then we've been finding that if we measure the diameter from the trees in the point cloud and then we measure them by hand, we see that they're almost exactly the same. So we go back out in the field in probably a couple weeks to do some post-glass fire measurements, and then we'll be out again this summer, and I'm looking forward to sharing more results with you in the future. Thanks.
Lisa, that's great. That looks like it's very scalable. Not only accurate, but scalable. So fantastic. We'll hope to see you next year up in Sacramento and see your results. Thank you. Our next speaker is Angel de Trinidad. He's a student at Chico State. The title of his talk is Spur Dynamics in Prune Trees. Angel.
Good morning. Next slide. So to begin, in many orchard crop systems, there's sort of been a transition from producers utilizing hand pruning to them using mechanical pruning. And that's because there's increased labor costs that are associated with hand pruning. And because of this, we're really sort of curious to see how mechanical box pruning impacts spur survival. As some of you know, spurs on trees serve as sort of the main fruiting body, which makes them so important. The health of these spurs are really dependent on a variety of factors, but potentially this box pruning method could be impacting some of those variables. The goal of our study is to essentially label and monitor spur health over the next couple of years. It's really similar to a study, as you can see here, done in Australia on almonds, but we want ours to be a little bit more comprehensive and holistic in prunes. We've labeled over hundreds of spurs and their characteristics such as number of leaves, number of fruit, the longest leaf length, to really be able to sort of track the spur characteristics and survivability over the next few years. Next slide. So for our preliminary results, we've really been able to look at the first trends in spur health. Firstly, we've been able to identify sort of non-destructive method in measuring the light environment, as seen on this graph here. By simply taking the number of leaves times the longest leaf length, we're able to analyze light environment without destroying the spur leaves that are so important. We've noticed the presence of fruit can potentially decrease leaf area, potentially creating a negative effect in spur survival. And lastly, we've also seen data that suggests that lower canopy specific leaf weight in the leaves in the lower canopy is decreased, which could potentially result in a decreased light environment. And that's important, especially because when you're comparing box pruning to hand pruning, box pruning could potentially cause a decreased light environment, which would impact spur survivability. Like I said previously, this study is going to be taking place over the next couple of years. So as more data becomes available, we'll be able to identify more trends and characteristics within the data. Thank you.
Angel, that's excellent data. It looks like, you know, who would have thought that the light environment is just really controlling so much. You know, the interesting thing about that is it might be amenable to artificial intelligence and hand pruning. So anyway, our next speaker is Sita Sisla. She's faculty at San Luis Obispo. The title of her talk is Evaluating the Utility and Consequences of Biodegradable Plastic Mulch from an Agroecological and Social Perspective. Sita.
Hi, everyone. Thanks for having us here today. So this is a very new project. It just started this summer and really this fall. So right now we're doing a new project looking at the consequences and utility of biodegradable plastic mulch as an alternative to conventional mulch. So next slide. So, so the problem is that a lot of specialty crops in California and elsewhere around the country and the world use plastic culture, so plastic embedding, in order to increase plant growth and reduce disease. And even with proper removal, we find that fragments are still left behind in the field, and over time these fragments can accumulate. Further, the plastic itself becomes part of the large global plastic waste stream. Next slide. And so a question that a number of researchers and agriculturalists are exploring around the country are trying to understand whether biodegradable plastic mulches are a viable alternative to traditional plastic culture. And so this is a picture from the field where biodegradable mulches are starting to break down in an herb system. So the goals of our project are to assess how agricultural plastic contamination affects soils. Next, test the efficacy of biodegradable mulch or BDM in field trials considering a variety of soil health metrics and plant productivity. Explore whether coupling compost addition plus BDM can accelerate breakdown, amplify productivity, and also increase soil carbon sequestration, and identify barriers to grower adoption of this technology. So that's the social side. And so one of the things that we've started with this project is just trying to understand how much plastic is in agricultural soils in California and what the effects are. And so we did some sampling this summer and fall, and we found that contamination in California agricultural fields can affect soil health. Click. Yep, and so this is just looking at microbial responses to different substrates in soils that are contaminated with plastics or not in agricultural fields. And what we found is that at both shallow and deeper depths in soils that are contaminated with plastic, there's a reduction in microbial activity and a reduction in the number of substrates that they can process. And so this suggests that plastic buildup from plastic culture in fields can have negative or deleterious effects on soil health. And we're going to continue to explore this throughout the course of the project. Thank you.
Great project there, Sita. Our next speaker is Mingxin Li. She's a student at San Luis Obispo. The title of her talk is Robotic Control of Yellow Star Thistle, Localization, Mapping, and Path Planning. Let's see, Mingxin, are you ready? Go ahead.
Yes, of course. So hello, everyone. Yellow star thistle, shown on the right, is an invasive weed that is very harmful to the environment and livestock. Unfortunately, there are shortcomings to all conventional methods for eliminating the weed. We propose an autonomous robot as a viable alternative. The control system of such robot, which was what I worked on, will have 3 essential steps. The first step, next please, selection of plants was actually not my focus because we have already trained the image recognition model using Google API. The second step, localization, the mapping, means finding the position of the plants relative to our robot. For this step, I used 2 different methods. The first one is by moving towards or away from the plant and use the mathematical relationship between apparent height from the camera, actual height of the plant, and changing distance to estimate the position. The second method works by moving lateral or perpendicular to the plant and use the change in angle and distance to estimate the position. We can also use the two methods at the same time to get more accurate calculations. The third step, path planning, is important because we want to find the shortest path to each location. The classical traveling salesperson problem, TSP, is very similar to what we're trying to do. Using TSP solving packages with minor modifications, I was able to find the optimal path to visit each plant. To conclude, the robots working will follow the scanning, mapping, and path planning process multiple times to minimize errors. Building the actual robot is more complicated, but the core control system involves the detection of yellow star thistle, the localization of mapping, and path planning. Thank you so much.
David, you might be on mute.
I am sorry. I turned mine off because I was carrying her previous broadcast. Sarah Caballero is a student at Pomona. The title is Stabilization of Bitter Orange Peel Byproduct by Binding to Plant-Based Protein and Pectin. Sarah.
All right, thank you for the introduction, Dr. Stowe. Next slide, please. So it is projected that this year, about 5 million tons of oranges will be produced in the United States, with California accounting for a little over half of this production. It is also estimated that about 3 million tons of oranges will be processed, and we are specifically looking at orange juicing. Next. When oranges are juiced, about 40 to 50% of the mass goes towards the actual juice. This means that about 50 to 60% is discarded as waste, or orange pomace. Orange pomace has multiple uses, including pectin and essential oils. However, one of the most underutilized uses, yet potentially very beneficial, are phenolic compounds. Next. Phenolic compounds have a variety of health benefits and would work very well when jointly with the million-dollar functional food industry. However, there are major roadblocks to their use in foods and beverages. For instance, they are not water soluble and they have very poor chemical stability. Next. We are addressing the challenges of citrus polyphenols by binding to pea protein. Our experimental approach is injecting a small amount of the polyphenol dissolved in ethanol into a pea protein solution, followed by vigorous stirring. Next. It can be seen from this image that the polyphenol in pure water had negligible solubility, whereas the polyphenol in pea protein had about 150 micrograms per milliliter solubility. This right now is about 60% encapsulation, but we are continuing investigation and believe that this can become higher. Once we determine the maximum amount of polyphenol that can be bound to pea protein, we will add another layer of pectin to these complexes to further increase their stability. Thank you.
Thank you, Sarah. Our next speaker is Siroj Pokharel, faculty at San Luis Obispo. The title of his talk is Antimicrobials and Reducing Foodborne Pathogens. Siroj.
Thank you, David and ARI for giving this opportunity. So can you please go to the next slide? So what is the problem here is we are facing a huge food safety all the time throughout the year. And we have the data saying almost more than 3,000 people, they died because of the foodborne food safety illnesses like infecting with Salmonella, E. coli, and Campi and all other pathogens that is from food sources. So our lab is investigating a type of bacteriophage, a virus that infect the foodborne pathogens. And the interesting fact about those viruses that you can see on the right-hand side of the image is it's eating bacterial lawn. The white lawn of bacteria is being eaten by the bacteriophage. So you can clearly see the picture. It's very white, you know, like there's nothing, no bacterial growth at the center. See, so this is being very active and against those pathogenic bacteria. So the thing is, it wasn't approved before, but now FDA and USDA has approved for so many pathogens, and there are so many bacteriophages they can act, you know, for the pathogens. And the other benefit is they do not act against our normal microbiome, which we carry all the time in our gut. Next slide, please. So how they work, so these bacteriophages they work in two different cycles, lytic or lysogenic. So the one that we're looking in our lab is lytic cycles, because they just incorporate their genetic material that you can see in the picture on top, and also they just disrupt the bacterial cell and make it dead. But the other cycles just take a long time, but because that incorporate the genetic material within the bacterial cell. Next slide, please. So one of the research that we did in our lab is we use lytic phases, and that was for Salmonella typhimurium. That is one of the pathogen that kills like people throughout the year because of the infection that may have in the produce or also in the meat sector. But I was focused in meat at this time. So there was a very good reduction of pathogenic bacteria. You can see in the 5% and 1%, even 1% lytic phases at the center with blue bar had almost more than 2 log of bacterial reduction in the meat surface. So the next goal in our lab, in our project, is I'm working on one of the ARI project looking at the marinated pork loins and its effect on Salmonella and E. coli on the different bacteriophages and its effect on those pathogens on the meat surface. Thank you.
Thank you, Saroj. Our next speaker is Todd Lone, a faculty at Fresno State. The title is Waiuli, a viable alternative rubber producing crop for poor quality soils in the west side of the San Joaquin Valley. Todd.
Thank you, David. I'd like to start out by recognizing one of our team, Colleen McMahon, who recently submitted some of our results for publication. Next, please. A problem on the west side is that during a drought, growers can lose water access to high quality surface water, which leads to increased pumping and the use of groundwater, which is high in salts, boron, and selenium. So our experimental approach has been three-phase. First, we evaluated the Waiuli lines for their tolerance to salt and boron in the greenhouse. And then we planted the best performers in field conditions. Secondly, we evaluated the effects of the salinity and boron on the quantity and quality of the latex and the resin produced by the plants. And finally, we're performing some economic analysis. Next, please. Key results, we've identified 3 Waiuli lines that have tolerated salinity and boron in the ranges that you see on the slide. Our analyses are also indicating that Waiuli does not accumulate the toxic ions of sodium, boron, and chloride. In terms of rubber and resin quantity, we're seeing rubber content in the 4 to 7% of dry weight range, and resin content in the 8 to 10% of dry weight range. Next, please. The natural rubber quality has turned out to be excellent. We're seeing no observed effects due to the high levels of salt and boron across any of the lines that we've tested in the field. In terms of the economics of the situation, right now, growers could be looking at producing a ton of rubber and 9 tons of material per acre. It is a drought, salt, and boron tolerant plant. It is relatively low on the input side of crops, and it is environmentally sustainable. Right now, we're estimating grower profit to be about $700 per acre, and that's obviously dependent on a lot of different factors. And I will conclude by thanking the ARI for funding this project.
Okay, perfect, Todd. Just by the way, is that a harvest at once and you're done with that plant, or can you do multiple harvests?
Right now, they're looking at anywhere from 2 to 3 harvests.
Okay, great. Thank you. Our next speaker is Kaomine Beng. He's also faculty member from Fresno State. The title of his talk is Organic Production of Agrati in Poor Quality Soils with Poor Quality Water. Kaomine.
Okay, go ahead and go on to the next slide. Basically, our research is basically a way to find if we can grow Agrati organically in California in poor quality soils with poor quality water. We like to use high mineral water because poor quality soil and water and sounds kind of like it's off-putting. So that's kind of what the term we've been using is high mineral water. Of course, California is continually in drought, and then followed by short periods of large rainfalls totals. So one thing that farmers are going into is utilizing poor quality water for irrigation and to grow these type of crop. Agretti is very high tolerant on salt and grown in Europe and Asia because of that. Right now there's 400 acres in the west side that is fallow because of high salt levels in the soil. So one of the ways to combat that is to grow a crop like Agretti, which absorbs selenium, boron, and the selenium. And again, those 400 acres have been there and haven't been produced on or used for 10, 20 years. So they're technically organic, and we can plant this there and use lower quality water and still produce a crop. Next slide. Our experiment, we had 3 goals. One was to find if we can grow it in the Central Valley with poor quality soil and water, and find out the effects of the water on the Agretti, and then evaluate the costs. Right there, you can see the students growing it in the west side of Fresno County. What we found is that Agretti can be grown here, and actually it grows better in high saline soils and with higher saline water. Next slide. So we actually grew more with the saltier the soil, the better the plants grew. We actually harvested and sold it in the Fresno State Market. We also took it to a couple of restaurants like Max's Restaurant in town and another one. And it got very positive reviews. The next steps that we're working on is to evaluate production costs and also do a market analysis and see where the interest lies. That's kind of the next step. One of the reasons why we didn't get to do some of the analysis was due to COVID-19. Everything, a lot of the labs were shut down, so we couldn't get some of the in-depth analysis. So thank you.
Perfect. Thank you, Caroline. Next year, bring some of that up to Sacramento so we can taste it. I don't believe I've ever seen it, or if I did, I didn't know it, and I certainly have never tasted it to the best of my knowledge. So thank you for that. So our next speaker is Chaitanya Muraka. She is a student from Fresno State University. The title of her talk is Quantifying Antioxidant Glutathione Levels in Tomato Leaves and Fruits. Chaitanya.
Hi, David. Thank you so much. Next slide. So ejection irrigation is nothing but the integration of subsurface drip irrigation and then the air component using a Ventura injector. So this is done to optimize the aeration levels in the root zone. The research mainly conducted in this irrigation had confirmed many positive influences on root growth, photosynthesis rate, and soil microbial activity. So we hypothesized that this happened because it helps in alleviating the stress in plants. Thus, we picked glutathione as a marker to indicate the oxidative stress. And we are comparing the levels of glutathione content in the leaf and fruit tissues to see if there's any difference between the aerated and the water only, which is the subsurface drip irrigation treatments. Next slide, please. Our experimental approach is very simple with completely randomized design with fruit treatments, and then the samples collected are stored at -20 degrees until the day of analysis. We use fluorometric acid test to extract and analyze the sample for the reduce and oxidize glutathione. Next sample, next slide. So the results are, in overall, the amount of glutathione is comparatively higher in fruits rather than the leaf tissues. And then it's evident that the amount of reduced glutathione is higher in ejection treatments when compared to the water treatments, whereas it's the opposite in the leaf tissues. So preliminary results indicate that there's a good amount of, it is a good amount of tool to assess the impact of ejection irrigation in order to reduce oxidative stress in tomatoes.
So thank you, Chaitanya. Our next speaker will be Eric Hauk. He's faculty member at Chico State. The title of his talk is Using Managed Aquifer Recharge with Surface Water Canals to Improve water management. Eric?
Great, thank you, David. As we heard this morning, a lot of focus on the Sustainable Groundwater Management Act of 2014. Most of the basins in the Central Valley are identified as critical or other priority basins that need to develop groundwater sustainability plans to prevent aquifer decline. California is also unique. We have the largest variability in precipitation. We could see most of the climate change models are expecting to result in even more variable weather conditions, which could lead to longer or more severe drought periods or flood periods. And the problem is we really need to figure out a way to capture and store excess flows when they're available. Next slide, please. A lot of times we think of capturing those excess flows behind dams, but as we also saw this morning, a lot of these groundwater sustainability plans are going to be based on capturing that water in the ground. Managed aquifer recharge, MAR, is a technique where you intentionally increase the amount of water that gets recharged into the aquifer as a way to store water. There's a lot of different examples available. The Kern Water Bank is probably one of the most famous. There, they use large recharge basins to distribute the water and let it infiltrate through the soil. There's projects in Arizona there where you can see large basins that are being used to put water on and have it infiltrate and recharge the aquifer. But a lot of times you don't have large open spaces available for these types of recharge basins. So people are starting to look at using existing farm fields. We can see down in the bottom left picture there, flooded up grape fields with it out of season, and some orchards there in Modesto, trying to use existing farm fields as a way to increase groundwater recharge. But there are some concerns that this activity could have an impact on yields and other plant health issues. And so our project, next slide please, was looking to use some of the existing infrastructure that's already available, and that is the surface water canals. Our partner is Orland Artois Water District, and they allowed us, there with a graduate student, Jim Norris, to dam up some of their canals when they weren't being utilized. And we were able to create some infrastructure there to fill those canals, measure the amount of water that was being applied into the canals, let the water sit in the canals and measure infiltration rates to get an idea how much of that water could be stored in the aquifer during the wet periods of time. We were able to get some really interesting measurements there in terms of how the water would transport through the soils. And we are currently using that data to run some models through the Central Valley hydrologic model to examine kind of what impacts these types of increased groundwater recharge projects could have on aquifer levels across the region. Thank you.
Thank you, Eric. Great work, very interesting. I would love to have follow-up questions. We've run a little bit over time. We might even say considerably over time. We had built in a break